Good afternoon. Today is November 5th, 2018. It is 2:56 in the afternoon. My name is Michael Gonzalez. This is for microbiology. And today we did a couple of experiments. But this is the first experiment for week 2 of our lab. As you see here, you're going to see 6 petri dishes of agar. And in each one there's a number. As you can see here, there's 6 of them. There they are. And we did a test tube for each one. So here we have a yeast. We have some yeast packets that we had to dilute some distilled water in them. And the distilled water, we added 2 milliliters of water into a test tube. And each test tube had the correlating number. So there was a 10-1, there was a 10-2, a 10-3, a 10-4, a 10-5, and a 10-6. In that dilution, when we made the mixture of yeast with warm water, tap water, we added 4 drops into each agar plate. And these are the results of them. I let mine grow for about 2.5 days, almost 3 days. It was 48 plus hours. And here we go. So this is the first one, T1. I don't know if you can see it. There is a lot of growth in here. We did the 4 drops. And there were so many colonies in here that I could not count them. And then here in T2, I think, or sorry, T2, in 10-2, we were able to grow more, and I was actually able to count these ones. In the sunlight, you can see them a little bit better. And I was able to count about 250. I think there might have been a little bit more, but this is the one I used for my report and for our variable. Here, you can see a little bit more. There was, I think, fewer than 30. As we got more diluted. In the test tubes, as we would dilute each specimen, of course, we get the primary from 10-1. We would do, if I remember right, it was 0.25 of ml into 2, and then 2 into 3, 3 into 4, 4 into 5, 5 into 6. So as we go further on here, here's number 4. It gets less and less growth. Same thing here in 5. Very minimal. You can see a couple here. And I think in 6 here we have nothing. We do have a little bit of a growth right here. But I don't believe that is the yeast. That might have been something else that had grown. But I was able to store these in the cabinet of my choosing. It was on average about 22 Celsius inside the room to allow the growth. But these are my results, as you see here. Their experiment was pretty interesting, especially for our first one. As we go on further, hopefully my technique gets a little bit better. But hopefully you can kind of see a little bit of our growth and development here. So thank you very much, and have a blessed day. Bye.